France, and he's going to talk to us about deciphering protein functional complexity with alternative splicing evolution.
Hi, my name is Diego Sea. I'm working at the Laboratory of Computational and Quantitative Biology at Sorbonne University. And I'm going to show you how to use alternative splicing evolution in order to understand protein functional complexity. So alternative splicing is a regulatory process that happens in most eukaryotic cells, and that leads to different protein isoforms that can differ in three-dimensional structure and also in function. We can imagine that each exon in a gene is like a train station in India, and if we are taking a trip, we can pass for some stations. So for example, if we want to go to Hora, we can start from Mumbai or Pune. So here we have two alternative stars. The same happens with transcripts. We can have also mutually exclusive stations and stations that can be skipped or not. And if we consider the entire trend line is very complex, at the same happens with considering different species and the set of transcripts for a gene in those species. So taking that input, we have several questions. What is how the alternative splicing impacts in the protein structures, and how this variation in the three-dimensional structure can impact in the protein interactions. Also, we want to know how alternative splicing leads to functional innovation, and which are the minimal evolutionary units of the alternative splicing evolution event. So to answer these questions, we developed two tools at our lab. One is Torrox. Torrox is a tool to disentangle orthology and paralogy relationship between exons, and Philosoph that allows you to reconstruct transcript phylogenies and also to model the three-dimensional structure of the protein isoforms. Both tools are freely available at GitHub, and they are implemented in Python 3. So the first tool, Torrox, has a pipeline that I'm going to show in this slide. Basically, the input is a gene name from any species you want, and a little species that you can give. And the tool automatically is going to retrieve all the transcript data from Ensemble for all the orthologous genes of the carry gene. Then the data is going to be cleaned up because there are, for example, incomplete transcripts and low quality sequences. So the tool automatically does the cleanup step before doing clustering of the exons in order to define cluster of putative homologous exons. These exons are further divided into minimal non-redundant units that we call subexons. These subexons, for example, can share the same sequence, but for example, here in the orange, we have two orange exons that share some sequence. So we define two subexons, one for the shared part, another for the part that is unique to the first exon. So we can represent each exon as a combination of subexons, and we concatenate these subexons taking into account the genomic coordinates and perform a multiple sequence alignment for all the species in the dataset. And this multiple sequence alignment is further analyzed in order to detect the orthologous exons. These orthologous exons are going to be the input for the following tool, that is Philosoph. And Philosoph takes as an input a gene tree and a list of transcripts represented by the orthologous exons. And basically it's going to give you a phylogenetic forest embedded in the gene tree, in which each tree of the forest represents the evolutionary history of a particular transcript. Also, Philosoph is going to use the protein isoform sequence annotated with the orthologous exons in order to search for structural templates that are going later to be used to reconstruct the structure by homology, and also to annotate exons and isoforms with structural features. But since a single iteration can lead to lower coverage, we iterate until we have templates for almost all the exons in the protein isoform. So coming from the set of transcripts in different species for a group of orthologous genes, we get three kinds of different outputs. The first one is the splice graph. The splice graph condenses all the information. Here, each node is an orthologous exon. And if there is at least one transcript in one species that has these two exons together, we draw a line. And the colors of the nodes and the lines indicates the conservation. So here, darker colors indicates more conserved exons and junctions. Then we have the protein structure in which we have each orthologous exon colored with different colors, and the transcript phylogenesis is generated by Phylosoph. So we apply this in a group of 16 protein families that comprise 15 human genes. From these 15 human genes, 28 shows conserved alternative splicing events. In total, we have 68 alternative splicing events that are conserved. We analyzed 900 transcripts, and each gene has between 8 and 91 orthologous exons. And we do some literature search, and we found that almost a half of the exons that participate in alternative splicing events that are conserved have an unknown structure, and more than 2/3 of the alternative splicing events that are conserved through evolution are also documented to be functionally important. So here we have 4 examples in which alternative splicing events is regulating protein interaction. In this case, it's protein interactions with DNA, RNA, with ligands, and also with other proteins. And for example, in the first example, when the exons that are shown in red change in between isoforms, what is changing is the DNA sequence specificity. That means that different protein isoforms bind different sequences in the DNA. But in the other cases, it's the affinity of the binding what is regulated. The last case is this one, is Tropomyosin alpha-1 chain. This is the splice graph for this protein. It's basically as the line train in India, but here we have the conservation information that allows us to see what are the alternative splicing events that are being conserved. So we can see 3 different alternative splicing events that are conserved through evolution. And if we look in literature, we found that these 3 alternative splicing events are known to regulate the dynamics of the filament of actin formation. This is an example of a kinase, CAMK2B. Here, all the alternative splicing is happening in the linker between the hub and the kinase domain. These alternative splicing are regulating 2 things. One of the things is the length of the linker, and it's known that the length of the linker is associated with the activation of the protein. And also there are some exons that are being alternative spliced that contains proline-rich regions that are known to be involved in protein interactions. Then in the case of myosin 1B, we can see only one alternative splicing event that is conserved. And if we look at the alternative splicing exon and we perform a database search using the multiple sequence alignment for this exon, we found other proteins that bind chymodulin, and in particular, we found EQ motifs that are being present in this exon, in particular at the beginning, at the end of a EQ motif that bind chymodulin. So the alternative splicing event is regulating the total number of the motifs in the protein and therefore the affinity. The last case is the Schenke protein family. Here we perform the reconstruction of the forest for the 3 proteins of the family. And one thing we can see is that, for example, the yellow transcript is not present in zebrafish because ensemble is under annotated, but we can use RNA-seq data to rescue these transcripts using the information of the phylogeny. So applications we can have for these two tools are, like in the last case, identifying isoforms in another organisms. Also, we can use it to do the search of the orthologous exon in order to identify those exons in under annotated genomes. We can use the structural models in order to understand the structural function of the exons. We can also use both the splice graph and the conservation and the phylogeny in order to understand the evolution of a given protein or gene family. And finally, we can combine all together in order to understand which functions are being related to this alternative splicing process. So I want to thank all the people in my lab that has been working in this project. Also at the INR for the funding of the project and the National Science Foundation for the travel fellowship to be here. And thank you all for your attention.
Any questions?
How do you take into consideration the exon length? Do you change it depending on the species, or do you have a procedure for formally modeling it?
Yes, we consider exon length during the definition of the subexons. In the definition of subexons, we can have, like in this example, two exons that differ in length but share some common part. So we are going to define a subexon for the part that is shared between them. But then the additions at the ends are considered as different subexons. And then you can see them, for example, in the splice graph. Sometimes you have some alternative exons that are before the shared part, so you can see that.
Any?
So some of the examples you showed that were like really powerful were a helix, and then some of the exampple you showed, when you showed them, they seemed to make structural sense. So it was like an insertion in a long helix of the coiled coil, or the insertion was in a loop. Can you think of any way to systematically think about whether or not the insertions you find are structurally compatible? I know it's a really hard question, and the answer is
yes.
no.
We are trying. Yes. We are trying in general to systematize this because a lot of these examples was handmade, basically. In these cases, we always found literature to support our hypothesis. But we are planning to annotate all the exons with structural features. So, for example, we can annotate disorder regions, secondary structure, length, and trying to make sense of those differences, but it's for the future. Thank you.
Yeah. All right. Thank you for your talk. I believe we understand that alternative splicing occurs within certain cellular contexts. So when you're checking it across different organisms, is that something you took into consideration? Thank you.
Sorry, I didn't understand the question. Context of what?
Cellular. So as in like certain cells would have certain exons being either expressed or spliced out and stuff like that. So was that also taken into consideration when you're doing the phylogenetic analysis?
The phylogenetic analysis is only taking into consideration the protein sequence and the definition of the orthologous exons, so we are not taking into consideration any particular characteristic of the transcript. I don't know if that answered the question. Thank you. Sorry.
Any further questions? Okay, so in that case, we'll thank Diego once again. We have a few minutes before the next talk starts, so if you haven't uploaded your slides and you have